Gamey Wright did.
So
Scott
did
is
you
a font
rotate those
of
in at all?
interesting
bit, yes.
trivia,
Yeah.
as well as
It's better off if you
phenomenal
don't, especially if there's any type of
biological
slope.
insight.
Because it gets really hot in the middle.
Well, it sure makes me want to go out and pulverize some black coal.
Sorry.
I shouldn't have said that.
For sure, you shouldn't have.
Yeah, that's much better.
What are these two white circular
Patty's
things?
Patty's been dying to get up to that little white donut up there.
Down to the
little white donut that's just
thing.
to the left.
What?
Of, yes,
I mean, is
what
this a little
that?
worm
power cycle
that's down below the surface that's created this? I don't see anybody coming out of the
Yes,
center.
I don't know.
Mesnio.
Yes, Nayo.
Yes,
I confirmed that with Tina yesterday that I was not yet. It's yes, not.
Yeah.
Yes, Nayo.
Yeah, but it has a negative connotation with the nya.
It means I don't know.
Right.
I am not knowing.
Right. Still, the nya in Russian is negative.
That's right.
Sorry, Patty, but this image is not really going to compete with the previous image of the Trisopathes.
Thanks,
you're
Chris.
right about that. Okay.
Okay, thanks, Pilot.
Pilot, I'm embarrassing myself. Let's go.
I was interested in it.
Thank you. Pilot says he was interested too. I appreciate that.
We always find
Just a little late in the
things
guy's humor, that's all.
Yeah, I know.
when we go,
It's okay.
that doesn't look quite right, and then we find something cool.
True.
That's right, you're right.
Stem?
I'm looking at these little white streaks that are coming down from the little sediment patches at the base of some of these layers. And I think they are just accumulations of 4M tests, shells.
And there's a very small white
Pilots,
coral
you thinking
in the
about a move?
background.
Ooh, look at this attachment structure.
I'm still looking pretty
And
straight
the
down.
bryozoan that goes with that.
I'm glad somebody zoomed in here because I've been telling you that you never see anything on the stem of Metallogorgia unless the tissue is missing.
Ah.
Look at that. The base of the skeleton, the tissue is missing, and there is
It's
the
like
brittle
crinoid.
scar.
Okay, thanks, Scott.
Push into the background.
That attachment looks not fibrous, but it looks like a,
like
a
cemented.
mesh, kind of.
Well, it's kind of cemented
but
down there.
It looks as if
I mean, it's not fibrous at all, for sure.
no, but it looks
Wanna go
looks
up the
like
stem
there's
and see
mesh.
if
It looks like
anybody
there are
else is on?
holes in it that you can see the black
And we'll just
through
pull
it.
off the top and keep going.
Possibly.
I'm having trouble hearing you, Rowan.
Like
Okay.
Possibly.
Talking
felt,
typical level.
sort of. Whoa. Beautiful.
Beautiful. Metallogorgia, really pretty.
Ooh. We're seeing so many of those. It's starting to get into my long-term memory.
Mine,
Time
we
we
can
have
get
left
a
on
ship.
bottom
we have about 50 minutes left.
270
5-0.
looks good for me. Maybe a little
5-0.
bit
Copy that. Thank you.
further
You're
south.
welcome. Plan wisely.
You do 260 if you want.
And zoom
Yes,
sparingly.
exactly.
Start picking out your candy.
260 co-pilot?
That looks
Don't-
good to
Dropper,
me, yes.
the dive will be ending in
30
50 minutes.
meters.
All right. Pick
Doesn't
out your
two
candy.
knots.
Don't wait too long. You may not get to it again.
Why don't you just do
Oh,
270?
look at all the little alluvial fans coming off the layers of sediment.
You don't really mean it that way,
No,
do you?
I don't. No, but golly,
270.
these layers are just
I
falling
know.
apart in front of our eyes.
Just
Ready to
enough
drive you now.
geology and sedimentary
We're ready
geology
for
to
a
be
ship move.
dangerous.
30 meters, bearing 270 degrees, decimal 2 knots.
Yeah, but these layers are not well indurated. They are just breaking apart, making
So
all
they-
this sediment.
Yeah, go ahead.
A really big, tall stock
That is
sponge
correct.
in the background upper right. Might be a little bit of a trip to get over there. But I don't think we've seen Calophacus on this dive yet.
You're right, Chris. We have not.
See how far I can get over.
Mm.
I think I have enough leash.
As your head
Yeah,
that way
yeah,
also,
if it means-
you
if it-
take left,
That are
so you
kind of
take
on
push
the
and
right
push
side
and
of it,
push.
too. Some smaller
Move
ones.
initiated. Good copy. Thank you, Bridge.
I could maybe give you a little more, but we're here.
No, I think I have enough. I still have enough. I'm just going up current, so it's taking a little while.
All right.
Yep.
Yeah, Josh, there might be something right over on the right, just to the right below your lasers also. Yeah, you're getting to it.
Yeah. So let's do this guy
We can look
first.
at both. Yeah, let's.
Come on in, Roland.
Chris was thinking this might be a younger version.
And there's something to the right of it.
Yeah,
on the
and even.
seafloor.
Yeah. Yep. Okay.
It's
Great.
saying, yeah, waving in the current.
Max zoom.
So this observation is always kind of interesting to me, to actually see what could have been the adult of this particular sponge off to the left.
Off to the left, right. Correct.
And if you were to measure the distance between the adult and the little child, or the keiki as we say in Hawaii, you're talking about, maybe a couple meters.
Yep.
And that would be my speculation, given that it's the only Caulophacus we've seen so far, but not really sure. Anyway, there is a sort of hypothesis going around that's circulating that some of these communities are created by a pioneer of a few individuals or single individuals from one species, and then they spread out from there. So that's the kind of thing I look for here. This could very well have been the parent of that one, but who knows?
Where's the other parent?
That's a good point.
I'm sure if we look around, we'll find one. Better be pretty close in these currents.
Well, I just don't know how these sponges work, actually.
Yeah, me neither.
I don't know if you know how the hexactinellids reproduce, or whether they can. They don't need two individuals to reproduce.
Yeah. I don't know, Chris.
Near the
Thank you,
smaller
Josh.
one, were there similar species on
There
the
were.
seafloor?
Yeah.
Very tiny, like maybe 5
Well, they were
centimeters
probably
or 3
like
centimeters.
attached to the bottom.
Yeah.
Different species.
Different species. Okay, like this guy.
Yeah.
Okay.
Yep. Yep, that's a different species.
What is it?
It's a glass sponge, a hexactinellid sponge as well.
All right, coming up.
Now, I have any sense of when that move should finish?
Wow.
Okay, if we can
Yeah, we're halfway
get
through it.
back over, heading west again, please.
So many.
And as you
Metallurgia.
do, I think it's off your rear left corner. Saw something very bushy. See it in
These rock
Sirius
layers are
as you're
just
backing up
falling
to it. Maybe
apart.
a spiral
Oh,
coil.
right behind you, off to port.
Now it's coming under you.
Ooh, there's a big white rock on the left.
You're over it
Or
now.
a big white
It's
patch.
port.
Yeah, I didn't want to hit it.
Yeah. Tether
Looks
looks
like
good.
white debris straight ahead.
Yeah, that's what Patty was just talking about, Ron.
That in a second.
There it
Oh.
Yeah.
Even
That's
locals.
the,
Mud.
Ooh.
right, that's the
very pinky.
Iridogorgia, I think.
That's a much more mature one.
It's a big one.
Coiled,
If you wanna
beautifully coiled.
Frame this, I'll do a pirouette around it and then go to the white rocks.
7, 8. That
Yeah,
has
want
this is
lasers
a
on?
really nice one. You can see this
Yeah.
is really big, and it looks like the coils are longer. Earlier, we were looking at one that seemed really tightly coiled, and so it's possible that those are two
Yeah,
different
can we get
species.
lasers
There is
on
one
the
that's
On?
called Iridogorgia
This is on.
magnispiralis, and that could be what this is. And earlier, we may have been seeing either Iridogorgia superba or splendens.
Right, and it is magna, all right. Look at those lasers. They're 10
Hey.
centimeters
Go ahead.
apart.
It's pretty splendid, too.
It is pretty splendid.
You got to give it that.
Yeah.
Wow.
I said earlier, these are among the most beautiful of the
Yeah.
corals in the deep sea, and they just never cease to make people go ooh and ah. Chris is pointing out that I did not mean superba. I meant Iridogorgia bella. Thank you, Chris.
And it is Bella.
Yeah. When you're ready, Roland.
It is bellissimo.
Yeah, you can see those names, Bella and Superba.
Partial.
Magnifica.
See a little amphipod on the end of one of those branches.
Really?
Look at the base.
Yeah, go ahead.
Looks like ship's done with the move.
Oh, brittle star at the very base.
We're fine as long as we want to spend here.
Look at that.
Oh, you can see little tiny sponge over on the left, too.
Oh, really? I saw it hair-like.
Little tiny.
Is that?
Look at that. What the heck is that?
Wow.
Oh my!
Is that a little tiny carnivorous sponge?
It
Could
looks like it.
Does look like it.
It has that sort of flat
Carnivorous pulling out.
appearance to the top of the thing.
Sure does.
Tiny
Wow.
though, tiny.
Yeah, but they were. Remember the other day when we got
Yeah.
them, Patty? They were little
Yeah, they
tiny
were.
things.
That's right.
You want to see the white rock?
Oh yes, please.
Okay, coming in.
We're gonna go take a look at this patch of
So
white
what
stuff.
is that pile of white stuff we're going to? That's pretty strange looking.
Well, that's why we're going there. We do not know.
Going into the unknown.
On our way, let me tell you that Les Watling published a paper along with Sonia Rowley and John Granat on the world's largest
Looks
known
foreign.
Gorgonian. It was a Rittigorgia magnispiralis from Twin Banks off the northwest Hawaiian Islands, and it
I
was
know what this is.
5.7 meters tall.
Wow.
This looks man-made.
Is that garbage?
Go ahead.
Oh, what is that?
It's trash.
It's trash.
That's what it is. It's trash. Oh, sorry, folks at home, but we
Looks like
never
part of
dive
a,
without seeing something
Small boat,
like
right? A
this.
ribbed boat. Maybe a rigid inflatable.
Is there a paddle, maybe?
So what do you think it
It's
is,
too big
Pilot?
for a paddle.
Too big for a paddle?
It's,
Pilot's speculating
fried
it's
I
eggs
was
somewhere.
suggesting
Paddle?
a rigid inflatable boat.
Originally
this
an
looks like
inflatable
aluminum
boat.
to me.
Rigid,
Yeah.
yeah.
Yes.
I mean, I'm.
Just the suggestion.
Well, maybe it's not aluminum. Is it fiberglass?
That looks like metal right there.
That's
That,
the rivet.
yeah, that's see the metal rivet.
Rivet.
Yep, yep. Yeah, you can see the
And is
rivets.
What's all that
What's
stuff on
on
the
it?
surface?
Is that
They're like
spray
demo
foam? Or is
Oh,
that sponge?
Looks like
it looks
sponge.
like demo
So
sponges.
is this airplane debris?
Oh, come on,
Don't,
Chris.
don't even go there.
No,
Lot of
please
ridges for an
don't.
airplane.
We'll never leave.
Looks like the underside of a boat with those ridges.
Yeah, that's what our co-pilot
Yeah, I
suggested
see
too.
that.
And you can see
Just like tilt
rivets
down. Look at the
in
foam
this.
or
So
that stuff growing down.
I hope that the occupants were okay. Little pieces of red. Sunken non-treasure.
All right.
It's been there a while.
It's covered with these, whatever these
Mm.
demo sponges are.
Right.
I know I'm 20 meters above you, but the surge or the heat.
well,
makes me
now we
nervous.
know.
Yeah, the camera makes you look like you're closer to.
Yeah.
Let's
It looks
see
like
the other side real quick.
some of them have dislodged and have anchored themselves, or maybe there was a little fragment from that.
Probably from one of those
Debris.
typhoons.
We have about another
something
40
happened
minutes.
Right
here.
above you. But we should be done with this move. Tether looks good.
It's a light blue section
It
underneath.
should have stopped 4.5 minutes ago.
Yeah.
Yeah, come on in, Roland.
Actually, I'm looking back at you, but the tether is still okay.
It's amazing how life will take advantage of things it finds on the ocean
Well.
floor.
All right, let's move on, I suppose.
Yep, that's fine.
Yeah. Come on out, Roland.
Did you put a drop point?
So my
into
guess
your-
is airplane debris because it's such a broken up piece, and typically airplanes, if they hit the surface violently, they can really break up into pieces like that. It's hard to imagine how a piece of a boat could wind up like that.
It's at star or something at the bottom of the screen, went off the bottom. Potential shrimp under lasers.
There's a kind of coiled whip.
Mm-hmm.
Another shrimp at the bottom. Something white
Quick zoom.
below that.
Is that barnacle come in?
Barnacle on a
Oh,
very
there's a
small
sea
piece.
star
and a barnacle.
and some little chunks of white stuff on the left.
You're out ahead of me again. We could probably start thinking about a move.
Sure.
Oh, that is the
Same
tiniest
heading as
star
Little
we've
good.
tiny
seen
little
so far.
star. Yeah, really tiny. A stalked barnacle.
Nice looking barnacle. Does have some
Got
sediment
crud.
stuck to
Got
it.
some crud on it.
Yeah, call it in. 30 meters, 270.
Sounds good.
Bridge, RV nav.
When you're good, video.
Ready for a ship move. 30 meters, bearing 270 degrees, decimal 2 knots.
We are coming to you live from the NOAA ship Okeanos Explorer. And we're
All these dark
broadcasting
spots.
from the Mariana
You see
Trench.
Yep,
National
come on in.
Marine Monument.
Video wants to know what are those black spots.
Probably old.
I think if we go
Bases.
back to the other thing that
Interesting.
we just
looking.
imaged, I think there's
Looks
a white
like it.
one of
Move
those.
Might be
initiated. Good copy.
manganese
Thank you, Bridge.
encrusted base of some critter.
Maybe
Maybe.
so.
Yeah, don't know.
My name is
So
Shirley
star on the
Pomponi. I'm
right.
from Harbor
I
Branch
agree
Ocean
with that,
Graphic
Patty.
And the
Institute.
left and
Yep.
the bottom.
Is that a
Little
four
coral.
legger under your laser to the left?
I'm the biology
Come on in.
science lead on this leg of this expedition. And we're looking at
A four-legged
Nope, there's
well,
a
you
piece
can-
of line
brittle
right
star.
there.
brittle star.
Oh, it's probably
somebody's
from
eaten
whatever
two of
it
his
is.
legs.
Possibly from that other thing we just saw.
Yeah.
And pteropod shell.
Yep.
That's the
And
little
what
white triangular
could
thing, right?
possibly be
Someone took a chunk out
a
of him.
sponge
Yeah, it looks like
spicule.
one got eaten.
ROV crew was thinking maybe a line of some sort, but it could also
that
Fishing
looks
line?
like
Is it a line?
monofilament to me.
Is
but
it
let me,
monofilament?
follow
Okay.
it. Let me follow it.
All
and,
right. Yeah, we definitely don't want to get.
Yeah.
Oh yeah, it's pretty long.
Oh, look
Oh,
at
yeah.
that. Yep, definitely.
Well,
Fishing
I don't
line.
know.
It is.
Right
Yes.
above and to the right lasers is something curved. Right at the foot.
Now,
Skid.
we get a chance to get an update on timing, what we need to do to make the waypoint,
Swimming
etc.
fish.
Okay, yeah, they want
Yeah,
get
go
the
along
fish.
west.
to follow the ridge.
Yeah,
Here
follow
we
the
go.
ridge.
And it is
there.
Yes.
I think Bruce may have
Yes, go ahead.
stepped
100 meters away.
out.
Okay.
You have 30 minutes left.
Oh, plenty of time.
Go ahead.
No, I'm still here. I just have the lag.
As you need.
Okay.
He's coming at me.
Yeah, he's coming at you faster than I can focus. You're full wide.
Oh, he felt my
Oh,
thrusters.
swimming away. Come back.
Go ahead, Roland. I mean, he's out of the. He's in my face, but.
Yeah, I'm at minimal focus. It's a quick
Yeah,
one.
he
You're
keeps-
full wide.
Sorry.
He's-
This
he's
is
I
our
lost
gone.
it.
first rat tail of the of this dive. We haven't seen one of these for a long time, I think. So it seems like rat tails are kind of rare in this area. But this looks like it could be the same little round-snouted
Did you lose your line?
fish we saw in the Hawaiian Islands
lose my
that Tomio
That fishing
Iwamoto,
line
I
you were
don't know
trailing.
where it
world
went.
authority on this group at the California Academy of Sciences, identifies as a Kumba species, Kumba. that generic name is an acronym of the Marine Biological Association of the United Kingdom, the initial
that star on the ground.
An acronym, I'm sorry. I'm forgetting the right word here. Anyway, mixing up the letters. So that was a rat tail or a grenadier. They're really hard to identify, even with extreme close-ups, but that's my best guess. They're usually a dominant group of fish at these depths, so I'm surprised that we haven't seen more of these.
I wonder if you'd see more of those, Bruce, if we were further in on this feature where there's more sediment, because although we were seeing them on very rocky terrain last year, so I guess that's not necessarily true.
Okay, nice,
yeah, and the other thing
nice
is
crinoid here.
That there may not be a lot of food for these down here. We're not seeing a lot of the small crustaceans and things
Josh,
that they
just
might
go up a
feed
little
on.
bit. I think there's a cup coral up there. Yeah, let's take a look at that.
That would provide a food source for those. So that doesn't really explain why they're so rare, and we're seeing the occasional halosaur or cusk eel. But as far as food that we're seeing in the habitats of this type could be one of the explanations.
What we're looking at right here is a cup coral, solitary coral.
And we're at 1610 meters, which is equivalent to the depth of our waypoint 2, 1609.
Can't tell if you are speaking.
Marshall.
Go ahead, Scott.
Oh, sorry, Shirley, you were breaking up a bit and I didn't want to interrupt. Yeah, so cup coral, you can see it's very transparent. This is a single polyp on the base of a skeleton. And when we were zoomed in, you could see little dots on the tentacles, and those would be where clusters of nematocysts, the stinging cells, are located.
Something bright orange right under you. See it
Black
on the
coral.
Yeah, sometimes the color's off of those minute cams.
Yeah, I guess it's not that
Come in
orange.
partial video.
It's a red.
It's
Yeah, I
pretty
see it.
red.
Come in partial. Okay.
Red crinoid.
Yeah, I see it, Roland. Is this of interest, watch lead?
No, we're good.
All right.
Continue on.
Come on out, video. We have a ship moved in?
Ship is just in position here.
Oh, right. Okay, so it's just finishing a move.
Yep.
Okay. Push north a little bit, see what we see. Let's come in on the sponge as I come by.
There you go again.
Very pretty.
Seeing more of these up here.
That looks kind of weird, huh?
It does.
So this is a Caliphacus, but it's a different one. And you got to think about this one for a bit, whether I've even seen something like this before.
Let's get the base.
Maxim.
It almost looks lobed.
Yeah, there's one species that's lobes like that, or subgenus that is, because the Caliphacus, most of them are undescribed. So I think this could be Caliphacus oxydiscus, but I was going to go check and confirm the subgenus.
Okay, thank you.
Full specimen.
Great. Thanks, Josh.
You're welcome.
Video's clear.
Come on up.
We had a crew change a little while ago now. I guess maybe about an hour ago, half an hour, about an hour ago. We have Josh, who's piloting.
Hello.
And we have David, who's co-pilot, and we have Levi on nav.
Right here.
Roland's over on video. And Dan is back here next to me, video clipping. They're all from the Global Foundation for Ocean Exploration. Part of our team of explorers out here on the ship.
That left of lasers. Little white.
Yeah, come in quick.
Okay, that's
small yellow something with a brittle star bigger than it is.
brittle star maybe on a hydroid.
Can we get close and look at that?
Does science want to look at this?
No, we're good. Thank you.
Thank you.
Guys ready for another move?
Yeah,
we could. I'm not
beautiful
sure if we're that much of a rush,
Iridogorgia.
right?
Yeah, we can get
Is
another
this
move in.
Magnabella?
You got
Same heading is good.
Cool. Sounds good.
This is Magna spiralis.
Magna spiralis. Sorry about that.
Bridge RV nav.
I think it's referring to very large spirals
We're ready for another ship
compared to
move.
Bella,
30
which has a
meters,
much more
bearing 270,
twisted
decimal 2
stock on
knots.
it.
Good copy, Bridge.
If they can. I don't know if they can.
Come on in, video. Okay, it's the crinoid we saw.
Another beautiful crinoid there. Okay, we have about
Move initiated. Thank you, Bridge.
about 25 minutes left.
Wow.
So Scott was trying to find out whether the Starapathes is a possible collectible. I wonder, before I say that, I wonder if we could zoom in on this stock crinoid that he's going down to right now.
That to me looks like it's a black coral branch, but I could be mistaken.
It's the one that's leaning
Yeah,
over.
Oh, I see the little one there.
Yeah.
Okay, yeah,
Is that a
all right. Okay.
Clamachlid crinoid?
I just have a black coral right at my toe, so I gotta move over a little bit. Don't want to hurt him or her.
Okay.
Both.
I think that there's some consensus that we would like to go ahead and collect the yellow Starapathes, the next yellow Starapathes that
Come on
we
in, video.
see. Everybody okay with that?
Show enough.
Okay, good.
Well, actually, can you describe what that is we're looking for?
Yes, I will describe. We are this is a crinoid that we saw earlier. What we're looking for, I'll be able to point out to you because we've seen quite a few of them. It's a fan. It's planar, mostly planar, yellow. I don't know, pinnate, planar. I'll show you. We've seen a lot of them.
Okay.
It's a black coral, the yellow black coral. Of course, we don't see any right now because I just said that. There may be one that we're coming right up on that's just to the right. No, wait, keep it. Go right.
Yep. Yeah, I'm going that way.
Okay.
I'm just looking left.
All right.
This guy? Or the next guy?
Okay, which one do you like better, guys? That one or the other one that's to the
Should we
left?
stop the ship?
Where am I? I'm way out. No,
You
we're going
are-
to need to keep moving the ship.
Okay.
Yeah, you're at 40 degrees.
Well, the bigger the better, always.
All right, bigger the
I'm-
better.
I'm
Let's go
gonna
for that one
follow
if you
you.
can,
My
Josh, the
heading,
one
I'm getting
that you're
thrown around.
close to
copy.
right now. Thanks.
I
Yeah,
would
just
say
go-
whatever one is easier for the pilot to grab.
Okay. Scott says whichever one is easier for you to grab, Josh.
Copy.
Ship is 15 out of 30
Yeah.
meters through the
And
boat.
actually, I have a really tight leash on you, so
Yeah,
Yeah,
we got to
we'll
wait
keep
for the ship to catch up with me here.
Yep.
But I will set up in the meantime.
I'm a little shallow, I think.
a note.
Yeah, you're
a
only
little,
at 10 meters.
Yeah,
Yep.
I know. It was 15 a second ago. We just came up with something.
We have a floating object in the port bio box. Do not open the port.
Yeah, do not open port.
Understood.
Glad somebody remembered that. Don't touch the port bio box. Starboard, starboard. Think
Copy that,
starboard.
Copilot.
we got
Think
it.
starboard,
Thanks.
David.
That's my note in all caps, big squares around it.
Oh, that's from when you were here.
Yeah. I'm going to change my heading to follow you.
Yep, that's fine.
I don't know how far ahead of us you are, but is it possible to, before getting this yellow, having a quick zoom on the white one that's just below the ledge? Right.
Okay, we're going to look. We'll see. Josh, is it
come on in, video.
possible to...
Yeah, no problem if you've already passed it. Don't worry.
No, they want...
I don't want.
Not,
This
I
guy?
don't
Not
think
this guy?
not that one. I think they want the
Come
one.
on out.
I think they want the one.
Come on out.
Want the one that's below,
Below
that.
the
Yeah,
ledge.
below the ledge.
Yep, coming down. Oh. This guy.
That little white one. That. I think so.
Am I tugging you around by the tether?
Come on in, video. Is your heading off?
No, that's okay. So
Yeah.
that's a crinoid.
Come on out.
So I don't think it was that one.
Okay, thanks, Shirley. Then we can move on to the collection.
Okay, let's go
I
ahead
know
and
the
collect,
current
Josh.
is spinning
Thank
me,
you.
but it's worse than it was a few minutes ago.
Yeah.
But as long as I'm not jerking around, it's fine.
I mean, I'm getting tugged, but like, you can come down a couple
Yeah.
meters, but not too much.
Yeah. I think we're heaving 5 meters.
5 meters, okay. Yeah, I wouldn't go any lower than that.
Yeah.
And keep an
So
eye on the
Patty's
tether.
there with you, is that right, Shirley?
Actually, she just stepped out of the room.
This feels better to me.
Okay, I had a geology question. It can wait.
You can ask me. I'll take a guess at it. No, I'm just kidding.
All right, let's zoom in on this, Roland. So we're going to want the whole thing, I'm assuming?
Yes, please.
Okay.
This has been among the most abundant of the corals that we've
You
seen
can come
today.
in tight. I don't see any associates.
I don't either.
All right, how's that for a zoom? Is that good?
Beautiful.
All right.
And there have been so many of
Come on
these,
out,
I
Rob.
would suggest just
Not
cutting
really,
it close
because
to the
it's,
base.
it's out
Yeah,
of focus and
yeah, that's what
Do
we're
you
gonna
wanna come
we're
back
gonna
in
try
and
and
get
do
it?
that.
Yeah.
Come back
Yeah,
in.
we've seen so many of these today. These are really abundant. So
It's
if you can
drifting
get it
so
as
much.
close enough to the base as possible, Josh, that would
Mostly
be
hasn't
great.
been in
Right,
focus.
copy.
You could probably dig it out. Right?
No, it's hard attached.
It's
Tina
on the
Molotova
rock.
says
on
Star
rock,
Pathies has not been documented from here.
Okay, let's get full specimen.
All right, let's get it with the lasers. The lasers should
Lasers
be on.
are on.
Come out a little bit more, Roland.
Yep, just gonna get a little bit more.
I bet it are.
You get it right at the base. It's better. Okay, pull on out.
Okay, I seem to be stationary.
All right, let's see if I can get a little closer to this. And then we're going to go in for the sample. It's taking a little longer to set up than usual
That's
just because
okay.
of the current.
That's okay. at 1,590. Wait, is that serious? Oh, no, wait, that's serious. Sorry. Where's my screen?
Both starboard bio boxes are
ready?
empty.
1,610.
Copy.
1,610 meters.
Dave? Oh, sorry. Starboard valve pack. So, port, I'm going to retract.
Yep.
Starboard extend. And do you want the drawer out now or later?
No, not now.
Okay. Pins are ready.
I like to give the drawer a close before you open it.
You take my camera, co-pilot?
Yes. What would you like me to do with it?
Tilt down a little bit. Yeah, hold that. How's that? Watch lead, is that
That's
enough?
fine. Yeah,
Okay.
that's fine. Thank you.
Okay, ready your drawer?
Okay. Opening.
Yep.
Coming out.
Alright, we're gonna have to try that again on a different guy. Sorry, watch lead.
It's okay.
Alright, you can close drawer.
Okay. Close on drawer.
There's one. Well, you see it. Just over to your left.
And retracting both pins.
Okay, yeah, I can go for that.
My position still looks good.
Yep.
That left one
You
looks
gonna-
as
you think
small
you're gonna
as
have to
what is
move?
left behind.
The left guy's a little smaller.
Yeah, okay.
I don't
Well,
know
we
if
can.
that matters.
You want to?
We can find a bigger one.
Okay, that's
Yep.
fine.
Right.
Okay, so that one floated out of the box. We're going to look for a bigger one.
I just lost. I'm going to give your camera back. I just lost you.
Yeah, sorry, I tilted your
Oh,
camera.
oh, that's what happened. Yep.
Yep, so if you tilt down.
Yep, I got you.
Alright, let's find another one of these. And then I'm going to have to stow it in a different way. Alright, I think I see another one up here.
Oh, hi, this is Bruce. I'm wondering
Can we
if
get
Scott
another
can tell
Yeah, we're gonna
us
need to
what
move.
makes these float. I wouldn't have expected that of a
240,
coral,
maybe
and I
20
assume
meters.
they don't have gas
Yeah, make it
in
30.
the tissues, so is it a high lipid content, or
Pretty
what
jarring
causes
now.
that?
Sorry, I didn't
Yeah,
copy
Bruce, I
that
was
watch lead.
just
Ready for a
typing
ship
an
move,
answer.
30
So,
meters.
these
bearing
black corals, the
240 decimal
skeleton
2
is
knots.
made of protein, so they're actually remarkably
Okay.
light. They're laid down in concentric rings and layers.
That is correct.
Maybe a bit of air space between them, not much, but there may be some. And there's so many of these feathery branches. I mean, the whole thing looks like a feather
this
that I
is gonna
think,
be a
you know,
big
in
target
part
right
it just
there.
kind of
Okay.
catches the
I'm probably going to pull you around until we
current
get closer.
that may be there. So ultimately, I think it's more the density and weight Some of these others, like the bamboo corals and certainly the stony corals, have a very heavy skeleton. There's
15
a lot
minutes,
more
so we got to be off by.
calcium impregnated along with the protein, and so they sink pretty heavily.
15 minutes.
Not
So we
those
gotta be
black
off by.
corals, not some of the chrysogorgiids.
Should be perfect timing.
Kind of variable. I don't know the question about the lipids. I suppose if there's a lot of eggs in there, if it's reproductive, there should be a fair amount of lipid, but I suspect, like you're saying, that's not really the answer.
Really comfortable going lower.
No, I know. Yep, come on in,
Thank you. That's interesting. Because a lot of the fishes at these depths don't rely on gas and gas bladders, but rather use liquid, light density oils in their tissues for buoyancy. So the interesting thing for me is that the rat tails are an example of a group that do sometimes have a gas bladder at these depths, and somehow they manage to keep the gas pressures in those bladders. At a pressure that works under the high-pressure environment, under this much water at these depths. And I'm not a physiologist, so I can't really explain that, but very often in the fishes, the lipids are the source of buoyancy for abyssal fishes.
Sure, Bruce, and that's similar to some of the scavenging amphipods as well. They have very large lipid droplets that seen to be used for buoyancy. There are certainly no gas spaces in the polyps of these corals, but I'm not sure to what extent they would use lipid as an energy store.
Come on in, video.
Okay, so it looks like we're getting some video of the small urchin before we do the collection of the black coral.
Yeah, you still need to move.
Okay.
Because every time I move closer, I'm turning your heading.
Sure.
Yep.
Ship has moved 15 meters.
So we'll be coming off the bottom in a little bit under 15 minutes, and for Shore-Based Science Team, we will have our dive planning call for tomorrow's dive, which will be both benthic and midwater. So we'll have our dive planning call for that dive in about 20 minutes, so about 5 minutes after we pull off of the bottom. This is a very tiny urchin that we've seen. Not sure if it's the same species, but it does look like the tiny urchin that we saw. Spines look a little bit smaller than the very long-spined one that we saw earlier.
Yeah, I think this is different also, Shirley, but I don't recognize what it is.
You turn your heading towards me a little bit.
Yes. My auto is right at you, but current and the tether are fighting me.
These black corals tend to be very slimy when you get them up on the surface. We collected one the other day, a small one, and it was
I was saying, where did that rock get its color,
slimy.
that rock-looking thing? It looks out of place.
Okay, come on in, Roland.
Now, it's interesting to me to note that this one that's in front of us has a lot fewer of the secondary branches, and I cannot explain why that is the case.
I think probably most of the ones we saw today, Scott, did not have the secondary branches.
I agree with you, Shirley. That's my impression as well. I think that one we were trying to collect before had quite a lot, and this one certainly has the secondary branching at the distal tip here, so that's fine. We will definitely get it on the collection. But it's just interesting to note how few there are lower down. And my recollection of the Storopathes storocrauda, the species that's on the Hawaiian slopes, even the very small ones have secondary branching.
What's wrong, co-pilot?
I'd love to hear from you, Chris. I was asking you in the chat room. I don't think you still have a question, but
the tether's okay.
In the Hurl archives, are you familiar with any yellow-colored Storopathes?
Yeah, it's golden colored. This looks somewhat similar to some that we imaged last year in the Northwest Hawaiian Islands, and the pictures are in the OER animal guide right now. One of the things that's sort of interesting is that down in American Samoa, we saw some colonies of this. I was annotating a guide for the Deep Coral Program, and we saw this thing, and all I could do is call it a branched Bathypathes. I didn't recognize it at all until last year when you and Daniel and Tina educated me on what these things look like. I realized that that's what we were looking at.
I'd like to monitor you.
I'm really surprised. You can see all the little bumps in the tentacles, which suggests to me, if I was looking at an anemone or stony coral, bundles of nematocysts, but I'm not used to seeing that sort of concentration in the black corals, unless I'm forgetting. So, Tina, if you could let us know on the event log, is it typical to see so many nematocysts on the tentacles of these black corals, these Storopathes?
Would you like a zoom?
Nope. Alright, so what I'm going to do is hold this, and I'm going to put my back into the current.
Okay.
So just keep an eye on my tether.
Yep, I'm watching.
Yep. Alright, if you want to open my box. Yep.
Retracting port. Extending starboard. And opening. Full open. Ready to close?
Okay, that's good. That's good. Good. Very nice. Very nice, pilot. Thank you.
Both pins.
That's okay. We got it.
Well, and there's the other piece. Loading away.
Yes, that's it.
All right.
Should I get rid of hydraulics?
Yeah.
Okay.
You can ramp down.
Valve packs off. Ramping down. And off. Okay. If you could head back north, that'd be great. I'll pick you up somewhere in between.
Got about 4 minutes. Pilots.
Okay.
Is that a different sea pen?
Not sure.
Come on in, video.
It's a crinoid. Oh, it is. CPN.
Yep, lovely.
Much longer stalk.
Very pretty. Very nice.
Sea pen with an amphipod?
Umbellula. Umbellula. Wow, pretty.
You are nose into the current, right?
Negative. Current's
Okay.
coming broadside to me.
Okay, that makes more sense.
Yep. Alright, come on out, video.
Tether's making weird shapes.
Yep. Current's coming out of the northeast. So.
Wow.
It's a big guy, isn't it?
This is so
Oh, look
Quite
how
5 meters,
beautiful that is.
Still pretty big. Okay, guys, get 2 minutes, so probably start. Time to.
So 2 minutes until we have to be on our way up, or 2 minutes plus some time to stow stuff?
That gives us a little bit of time to stow stuff.
Okay.
Okay, so we have about a couple minutes left in the dive.
And when do you want to do the
We
post dive
At
discussion?
about 7 minutes.
Okay.
We could go.
Yeah, go ahead.
We could go take a look at that sponge by the lasers if we had a couple minutes.
We could,
Yeah.
and we shall.
All right. And once again,
Just want to confirm this is not Caulophacus. I think it's Bolosoma. But just wanted to get a better look. I thought I saw one earlier.
The one that looked like it was kind of lobed.
No,
that was a
this is
different
a different genus altogether, and even a different family.
yeah,
The
yeah.
Bolosoma is euplectellid, and the Caulophacus is whatchamacallit, or Zelid. Yeah, this is definitely Bolosoma.
Huh. And that's a euplectellid?
I was typing. Sorry.
Go ahead, Scott.
I was just going to ask, you guys were talking about the sponge. I don't know if you noticed, it looked like one of those scoop sponges farther in the distance. Semperella or something like that. I forget the genus name.
That's the name, Scott. I didn't see it. That's pretty interesting.
Hey,
Yeah, it's when we zoom back out, you'll see it's in the distance. But it looks pretty clear. That's what it was.
Okay, pilots, it's time to back up.
We can move on.
So one thing you'll notice that's different from this and the Caulophacus that we've seen for several days is that big, huge osculum on top. And if you spin that guy, oh, I see what sponge you're talking about, Scott, off there to the upper right. Anyway, if he's coming around here, and when you take a look at the other face, you'll see a big, huge scooped out part. And so they're very distinctive once you get a good look at them.
Easy.
Sorry.
Okay. Come on in, video. Go ahead, video. Well, we stow our swing arms.
Yeah,
Oh, yeah.
You nailed it, Scott. Semperella for sure.
Yep, and you can see that really nice spicule brush at the base.
Teaching me something here.
It's like sediment
Securing hydraulics.
cowled around it.
So again, I sort of preach this every time we take a look at the base, that you can see how different the attachment is on this sponge versus that dead stock we took a look at a little while ago. So this is very, very fibrous, this attachment, and very well suited for anchoring sponges in the sediment. So a lot of the sediment-dwelling sponges actually have this type of fibrous attachment.
And
very cool. It also looks like there's been a lot of scouring so that it's on a mound in a depression.
A turn in.
To me.
Yes.
Yep, and I can get rid of that.
All right.
And Scott, when you say, or not Scott, Chris, when you say fibrous attachment, those are actually spicules, right?
Yeah, that's correct. Yeah, and it's the technical term is lophophytus attachment there, which is just loose, unglued together spicules, if you will.
Cool. Very rippled. Yeah. Oh, Scott just wrote that down. Rippled surface. As we're getting ready to leave the bottom. Vehicles have starting to get everything tucked away so they can come off the bottom. This is
something. The current's going to be pushing us this way.
Okay.
So I'd keep your heading that right about there.
So this has been a really interesting dive. We've seen very. We started out at 1,914 meters, ended up at 1,607 meters. Very interesting geology that I think may have had our geology experts stumped about what exactly we were seeing.
That's for sure.
Beautiful, beautiful layering and stair steps. Right. The biology has been very, very interesting. We're back in coral land.
Yeah.
So we saw some really abundant metallogorgias. We collected actually 2 corals today. We collected an absolutely beautiful Chrysogorgia, and then the Stauropathes that we just collected as well.
Okay.
So we've seen a camouflaged isopod, very cool. A sorcerer eel.
Come up slow.
It's just going to take me a little bit to
tons of stuff. A Halosaurus. Lots of bamboo, probably not lots, but a lot more than we said, relatively speaking, bamboo coral as well.
I would say there were a lot, Shirley.
Yeah? Okay, good.
Umbellula sepens.
Yep. Several different species of hexactinellid sponges, and now we're starting to take them for granted, the carnivorous sponges, because we're seeing them all the time.
They're everywhere.
So, rat tail. Lovely Iridogorgia. My gosh, really large samples of that, individuals of that. And we finished up looking at a Semperella with a fibrous spicule attachment. We are going to be diving tomorrow at a place that we've called Subducting Geo 1.
Right.
And it's going to be it's a deep dive. It's going to be a combination of a benthic dive, so we'll be exploring along the bottom. And we will also be concluding that with a 2-hour mid-water dive, which should be very exciting because we're going to be going back. Series of transects at different depths. Should be really, really exciting. So for those of you at home, please tune in tomorrow. The dive will start a little bit later because we're diving deep. We're going so deep. So please tune in tomorrow, and thanks for joining us today. We are going to sign off now from the public feed and
wave to everybody, Shirley.
Bye.
See you tomorrow.
Bye, Mom.
Bye, Gerard and Catherine.
Mom doesn't have a computer. She can't see me. And we're going to have our dive planning call in just a couple of minutes. So for those of you shore-based science team, if you can please just stick it out with us for just a few more minutes, and we will do our dive planning call for tomorrow's dive.
31, I think, is the max you want to do, because otherwise I got to